So I think this is an ion channel, probably one of the last great families of ion channels to be discovered. It's actually quite a small family, just two members. It wasn't discovered by us. It was originally reported as a FAM38A protein in 2006, and actually Sophion to be linked to RAS signaling. But it's best known because of this paper from Ardem Pataputian's group and Bertrand Coste, linking it to mechanosensation. And this is true. It's a fantastic mechanosensor, and you'll hear more about this from Gary's talk as well. So these proteins assemble as trimers. It was initially thought they might be tetramers, but they're trimers to form large channels, approximately 25 picosiemens. They're mixed non-selective cationic channels, sodium and calcium permeable channels. This is from Rod McKinnon's recent structural paper. There are a couple of others published just after this, all around the Christmas time, showing similar structures following on from the original lower resolution structure here. So really quite a beautiful ion channel, this tri-blade propeller-like structure shown here, more in the ribbon structure, and then in cross-section here. And Rod McKinnon suggests that it causes slight curvature in the membrane. This might be important for how it senses membrane tension. All of the data are indicating that it's a direct sensor of membrane tension. There are a couple of experiments here from one of the publications suggesting this from Walter Grandel's group, studying the channel either in inside-out patches or outside-out patches and applying stretch or negative pressure to either of these patches, which obviously in the opposite orientation you get channel activation. So it seems that just an increase in membrane tension is enough, and it's quite a rapid effect. This is a millisecond timescale. You see some inactivation here. In many of the publications you see a much faster inactivation, and I'm going to show a non-inactivating state later on. So this is quite an interesting feature of the channel. It seems to be context dependent. So I'm head of a cardiovascular institute at Leeds, and so obviously we're interested in cardiovascular disease and in the worldwide epidemic of diabetes and obesity, etc. This is a major issue, and roughly half of people are dying or being disabled from cardiovascular disease. And a key feature of the cardiovascular system is its ability to sense blood flow. And all of these beautiful vasculatures you get in the embryo, the placenta, or the heart, or the brain, or the extremities, and some of the vasculatures we don't want, like in cancer, these all depend on the sensing of fluid flow. So it's a bit like a road traffic system. If the roads are used, then we keep the roads. If we don't use the roads, then we might close them down. And it's the same in the biological system. So the sensing of fluid flow is critical in biology. And you'll see later on, if you don't sense the flow, actually you don't get mammalian life, at least. And in the adult, it's thought that the sensing of flow, so it's happening in us all the time, is necessary for the healthy state of our vasculature. But the fluid flow is very complicated. This is one of the 4D MRI studies from the Leeds group, which specializes in MRI a lot. So this is with abnormalities in the aortic valve causing disturbance of flow, as you can see by these different colored lines. But of course, the vascular system is highly complex and the fluid flow is going all over the place, and exactly what fluid flow there is matters for the healthy state of the blood vessels. And the cardiovascular disease, most common one, atherosclerosis, is occurring in areas of disturbed or low shear stress or fluid flow. So what we're really interested in is how this is sensed. And so these are a few quotations from key players in the shear stress sensing field, an American, Australian, and German, all saying that actually there's not much consensus about that, and that's actually still the case. So quite a big challenge actually to work this out. So I think a key observation, not made by us, was from Joachim Hoyer's group, published in 2002, using cultured endothelial cells, where they observed, using patch clamp recording, approximately 25 picosiemen channel. It was a non-selective cationic channel activated by fluid flow in these cells, and the molecular basis was unknown, and we were very interested in that. Some of you might know that I've worked for a long time on TRPC channels, and there have been suggestions that they are mechanosensitive channels, and they're expressed in endothelial cells, etc., but we could never reconstitute that. We didn't publish those data, but we could never produce anything very convincing there. So we were interested in the original report from Ardem Pataputian, which was in N2 neuroblastoma cells, and whether it might be relevant to the endothelial cell. And because the piezo channel is 25 picosiemens, one from Joachim Hoyer's lab in endothelial cells was 25 picosiemens, we started to look at this as soon as Ardham's paper came out. So back in 2011, although we didn't publish till 2014, we were studying these human umbilical vein endothelial cells. It's a common preparation for studying endothelial cells. It's an unusual vein, actually, because it's oxygenated, providing the mother's oxygen to the baby. Anyway, it's a convenient way to look at human endothelial cells. And we found that the piezo-1 protein is expressed quite nicely in these cells. We used 3 different siRNAs to knock it down. The antibody is not specific, and there's still a problem for the field. But we made cell-attached patch recordings, applied this suction, and we managed to activate channels, not quite as rapid as had been seen for overexpressed piezo, but nevertheless the conductance was right at 25 picosiemens, and when we knocked down piezo, we could reduce the frequency of this. We've since done quite a lot of recordings on freshly isolated endothelium, because there's a lot of worry about cultured cells, of course. I'll just show you one example. We're just trying to publish these data at the moment, but it's work on human placenta, and we're dissecting out arteries from the placenta obtained from the hospital just next door, and then sort of taking a little bit of artery here, and then freshly isolating endothelial cells and studying them immediately. And what we're doing then is excising patches, pulling off outside-out patches, and applying fluid flow. And what's quite nice, and I'll show you better data on this later on from the mouse, is that if you apply the fluid flow, then you activate an ion channel, which is 26 picosiemens, so very similar, and inhibited by gadolinium. You'll maybe know from the piezo field the blockers are not very good, so we've got gadolinium, ruthenium red, but it's one of the agents that you can use. And I'll show you later on genetic data, in the mouse at least, to say that this is piezo-1. So this suggests that the piezo-1 channels are in the endothelial cells natively, here relevant to the human, and that they are directly, or at least in a membrane delimited fashion, activated by fluid flow, so mechanically activated by this physiological force. So I'm going to talk to you about this vascular aspect of piezo-1, and particularly about these things in bold. But just to mention that piezo-1 is quite broadly expressed, does many different things, and so there are quite a lot of other things that have been reported in the literature if you want to have a look at that. There are genetic studies suggesting links to lymphatic development and non-immune hydrops fetalis, also links to anemia through genetic studies, and a beautiful study by Ardham's group recently about malarial protection, and a variety of other things as well. So we were interested in, obviously, this fluid flow phenomenon. As I told you, you don't get mammalian vasculature unless you have fluid flow, and so a critical time in development is just after the heart starts to beat. So the endothelial cells have already formed a nascent network, sort of a tubular-like structure, but it doesn't mature unless the heart starts to beat and push fluid into the network. So the heart starts to beat in the mouse around about day 8.5, and so we're looking just after that. And this is the time when you start to get vascular maturation. So like as I said before, if the cars start to flow on the road, then you start to get bigger roads. If the blood starts to flow, you get bigger vessels. So you can see an example of that happening here. This is the nascent network of endothelial cells. And then as the heart starts to beat, then you start to get vessel maturation, allows blood flow to the developing organs, and you get progression. So we isolated endothelial cells from these embryos and applied shear stress, the fluid flow, and we could activate calcium entry. So this is one of the characteristics of the fluid flow response, an early calcium event. And if we knocked out piezo-1, and then there was nothing at all, but you could still get a VEGF response. Another fluid flow response you can see is alignment of the endothelial cells to the direction of the fluid flow. And if you knock out piezo-1, then the cells are all over the place. And if you look in the animal itself, basically the homozygote knockout is lethal. So you have to look at early in embryogenesis. So we're looking at day 9.5 here, also at 10.5. You can still see some embryos. But if you look carefully at the vasculature, in the wild type, as I say, you get these maturing vessels. But in the endothelial-specific deletion, and it's also true in the global deletion, they're about the same, then you can still see green staining for the endothelial marker, but the endothelial cells are quite chaotic, and you don't get this organized vasculature. So the basic idea is as the heart starts to beat, then the blood pushes into this early forming endothelial network. The flow is sensed. We think it's sensed by piezo-1, and that allows development of blood vessels, organ expansion, and the embryo goes on and you get birth, etc. But in the piezo deletion, then vasculature doesn't mature, you get growth restriction and resorption, you don't get any live births. So it's necessary. So I think this is quite a good test of whether something is a shear stress sensor in the vasculature, because if you look at this literature, there are many candidate proteins And not many of them are lethal at this time point. We're interested in what the downstream pathway might be, because if you sense the fluid flow, then how do you get the reorganization of the vessel? Well, of course, we don't really know, but I'll just give you a little bit of insight here. So we've done a proteomic approach where we've compared cells that have wild-type expression or with knockdown piezo, or we've also done the knockout here, and then done phosphoproteomics, titanium dioxide trapping to pull out the phosphorylated proteins. We also see other proteins, and then look to see which things are related to piezo. And one thing we found was that pretty much all of the downstream substrates of calpain were downregulated in the piezo-1 knockout, suggesting some link between piezo-1 and calpain signaling. And so we looked at that. We looked at calpain activity, and we found that in the knockout, the calpain activity was very much reduced at these 2 embryonic days. We did some cell culture experiments where we applied shear stress, and we could activate calpain and block that with a toxin which inhibits the piezo channels. We could see the piezo channels labeled with GFP at the back end of the cell in the focal adhesions, shown by this green labeling here. And then as we applied shear stress, then the focal adhesions and the piezo, because this is green is the piezo, disappear. So our idea was that the shear stress is applied, it activates the channel, gives calcium entry, calpain activation, and then that essentially digests part of the focal adhesions, releases them a bit, so the cells are then free to start moving and rearranging and getting vessel maturation. And we did some tests of that with calpain inhibitors, and it worked also, calcium-free solution, etc. So that seems to be part of the story, at least. As cardiovascular biologists, we're really interested in what happens in the adult stage, and we've just started that work, and it's a long process. But I'll tell you a little bit about where we're up to so far. So of course we're doing human studies, but I'm going to show you a mouse study here. So this is where we've done a genetic deletion of piezo-1 in the adult, because if you delete piezo-1 globally, it's just lethal, so there are no live animals. So we're doing a conditional deletion, so it's a tamoxifen-induced deletion using this V-cadherin or CAD5. This is an endothelial promoter, Cree deletion, from Ralph Adams' lab. It's a very commonly used system in the cardiovascular field. And the first thing you notice is that the control mice and the endothelial deletion mice look basically the same, yeah? And we've actually done quite a lot of general cardiovascular phenotyping of these mice in this the condition where they're just in the cage running around, and they seem to be pretty normal. I haven't shown you all those data, but we have published them and you could have a look. But to show you a little bit more of no difference, no effect, this is a contraction study. Actually on the mesenteric arteries, so of course they provide blood to the intestine, and it's a conventional old-school contraction study. So what we're doing is applying phenylephrine, agonist alpha-1 adrenoceptors causing contraction, and then relaxing with acetylcholine acting on muscarinic receptors in the endothelium to cause vasodilation. And then you see this relaxation occurring here. If we delete the endothelium, then you don't get much of that response. So this is a wild-type animal, and this is classical physiology. In the piezo-1 endothelial deletion, what do you see? Basically, you still have a phenylephrine contraction, you have an acetylcholine-induced relaxation, and it's endothelium-dependent. These are the main data for the wild type, and for the piezo knockout, looks very similar. But we got an impression here that maybe the acetylcholine response was a bit larger in this case compared with this one, but statistically not demonstrated. So, but we were suspicious that something else might be going on, and hopefully I'll convince you of that. And I think this ultimately will turn out to be very interesting. For those who know a little bit about the endothelium-induced relaxation, as this acetylcholine response, they'll know that it's quite complicated, and this has also been quite controversial how this works. One aspect of it is through nitric oxide release. Another part of it is through something called endothelium-derived hyperpolarization, and there are some studies suggesting there might be a factor as well, and it has variously been called EDHF or EDH. But I think what's of relevance here is the EDH bit, and you'll see why hopefully later on. This hyperpolarization in the endothelium arises because of potassium channel activation, and you need to activate two different potassium channels, SK and IK. And the SK channel can be blocked by apamine, and the IK channel, intermediate conductance, blocked by charybdotoxin. So this is now a classical experiment in vascular physiology, that if you block both of these potassium channels together, you can't do it with one or the other, both together, then you block this EDH mechanism. So we've done this experiment, and this is in the wild type first of all. We'll look at the acetylcholine-induced relaxation, and then with the toxin, and you get a slight decrease, suggesting a small EDH component. Then when we look in the piezo knockout, so all the data are down here, original trace over on the left, here you get the acetylcholine-induced relaxation, but in the presence of the toxins, now there's almost no response to acetylcholine. And the mean data, hopefully you can see, this is the control response to acetylcholine, but now in the toxins, there's a much bigger effect compared with the control mice, suggesting that the EDH component now is bigger in the piezo 1 knockout group, suggesting that piezo 1 is anti-EDH. So if you get rid of piezo 1, you get more EDH. And that is confirmed here. So we looked then more carefully at this. So the experiment I've just shown you there is actually a static experiment, but I'm talking to you about fluid flow. So then we want to do fluid flow experiments. So what we did was we freshly isolated endothelium from these second-order mesenteric arteries, and we applied fluid flow. We're measuring membrane potential. The cells sit around minus, so it's a sheet of endothelial cells, freshly isolated. And then we're measuring membrane potential around about minus 43 millivolts, the mean data are down here. And we apply flow, and you get a depolarization that's consistent with activation of piezo 1 channels, because they're non-selective cation channels, so they cause depolarization. If we do the piezo knockout here, first of all, we see that the resting potential is more negative. The mean data are down here, so significantly more negative. And now the fluid flow, if anything, goes the other way, and you don't get the depolarization. So that tells us two things. One is that the piezo 1 channels are constitutively active, and secondly, that they are responsible for a depolarization caused by flow. What we did then was we then excised membrane patches, as I showed you before, from this endothelium. So we're pulling off little bits of membrane to see what's going on with these channels. And the first thing you see is that there is spontaneous activity of the channels. And if you look in closer resolution, you can see the unitary events. The conductance is around about 25 picosiemens. In the piezo knockout, there's absolutely nothing at all. Mean data over here. So these are piezo 1 channels. If we apply fluid flow to this excised membrane patch, then we can activate the channels, and then we can inhibit with gadolinium. So here in this very restricted environment of the outside-out patch, we are getting fluid flow activation of piezo channels in a sustained kind of manner. So this actually contrasts with a lot of overexpression data suggesting these channels inactivate completely. I think Gary will talk more about this later on, but these channels can clearly have a non-inactivating state, and this is obviously important for vascular physiology, which is a slow kind of business. So what we're interested in is then how does that work and relate to the physiology? So in the endothelium, we've got these channels which are partly constitutively active and then can be further flow activated. They give sodium-calcium entry. This is a depolarizing effect, so that will be anti-EDHF, which is endothelium-derived hyperpolarization, which is a relaxant effect. So if you're anti-EDH, you'll get contraction, so that should be the case. So we did essentially a dose-response curve for flow against membrane potential, and as you increase the flow, you get more and more depolarization. In the endothelial knockout, nothing. We then isolated vascular smooth muscle cells to look at the threshold for activation of voltage-gated calcium channels, which are necessary for the contraction. Looked very carefully at that. These are very small currents, but these are absolutely essential for the contraction. And then plotted the data. And this is the shear stress evoked depolarization, and this is the activation of the voltage-gated calcium channels. So the idea is that if you depolarize increasingly in the endothelium, that's transmitted to the smooth muscle cells via gap junctions, and there's a lot of literature on that. So these are electrically connected. That then depolarizes the vascular smooth muscle, just sufficient to activate the voltage-gated calcium channels and cause contraction. So is that really true? So then we cannulated arteries and applied flow to them. So this is one of these arteries from the mesenteric bed, and then we've applied flow by putting a pressure differential across the blood vessel. So the pressure in the middle stays the same, but now there's fluid flow down the middle. And hopefully you can see that the diameter of the vessel gets smaller, and you can see the constriction occurring here. This is then blocked by nicardipine, which blocks voltage-gated calcium channels. So this is it's consistent with a depolarizing effect to activate these calcium channels. And strikingly, in the piezo knockout, then there's no constriction at all. So this is a knockout in the endothelial cells. So this is a transmission of electrical signal, a depolarizing signal, to the smooth muscle cells to activate voltage-gated calcium channels, which is lacking in the knockout. Now, is that relevant in vivo? So we then made telemetry measurements of blood pressure from these mice. And it looks kind of complicated, but this is the mean blood pressure. This is the systolic blood pressure, and this is the diastolic. And across the top is when the mice are active on a running wheel, and across the bottom is when they're inactive. And hopefully you can see a clear pattern that in the active one, when they're running on the wheel, there's a big difference between the control and the piezo-1 endothelial knockout. But when they're inactive, there's not much difference at all. So what happens when the mice are running on a wheel and they're active is you get an increase in blood pressure, and that's piezo-1 dependent. That suggests that this vasoconstrictor effect is occurring in exercise. So something we'd seen in vitro in the organ bath now is showing itself in the animals, but only in a particular condition, and that is when the animals are actively running, which of course is when you've got increased blood flow. This is a picture you can find on Google just to find out what happens when you're exercising. And so normally, if you're just sitting around, then a lot of your blood flow actually goes to the intestines, and that's where the mesenteric arteries are. But if you start to exercise, then the blood flow decreases strongly in the intestines from about a quarter down to about 5%. And the blood flow to the skeletal muscle increases, and obviously it's relatively low when you're sitting around like we are now. So there's a redistribution of blood flow, so a constriction in the mesentery. But what happens in the skeletal muscle, because you don't want blood vessels constricting in the skeletal muscle, you want the blood flow to increase here so you can run around. So we also had a look at blood vessels that go to the skeletal muscle. This is to the leg of the mouse. And this is one of the arteries, a saphenous artery. It's quite difficult to do in the mouse because they're all so small. But basically we looked to what is the EDHF component in these arteries. This is the control, and then this is the knockout. And basically it's very similar in the control and in the knockout. So in the mesenteric vessels, we saw an anti-EDHF effect of piezo-1, but in the saphenous artery, then we don't see that effect. So it suggests a vascular bed specific effect of the piezo-1. So what we thought was this could affect the performance of the animals, and we've looked at this. This is what we've published so far, but I think this all gets very interesting, I think. That then looking at how well the animals can run around on the wheel. Because obviously if a redistribution of blood flow is important, and then you can't redistribute it, then presumably you can't perform as well. And that turns out to be the case, although there's some adaptation, which you'll see. But if I can focus you, first of all, on the first night of exercise, when they first get exposure to the wheel. So before they've just been sitting in the cage, and now they get a wheel, and they're very excited, and they run a lot on this wheel. And so you see in the control, this is the number of activity bouts, just over 40, but in the knockout, it's only about 30. It's the time active here, it's also less in the knockout, and then the distance run, normally running around 2.7 kilometers a night, and then down to about 1.7 kilometers in the night. So the performance is less. But then you'll have noticed that then gradually over the night of training, etc., then there's some kind of adaptation and the effect is lost. So how that works, we don't know. This is what we're studying at the moment. But anyway, there's a decrease in performance in the endothelial knockout. We also looked at the body weight of these animals. And normally, so the 12 days after they've had the tamoxifen injection, they gradually put on weight, because they're just sort of wandering around the cage and eating, etc. But then when they get access to the wheel, then the control mice no longer gain any weight. So it's just like us when we're exercising, then we can attenuate our weight gain. But in the piezo knockouts, they're actually about 2% less body weight, so actually significantly less. So within a week, they're losing about 2% of their body weight. So that would be equivalent to me losing about 2 kilograms in weight in a week, which I'd be pretty pleased with, yeah? But it would be good, yeah? So it's actually quite a lot, although it looks a small effect, it's quite significant. So this is physiology we're dealing with. So we're really interested in the idea that maybe piezo-1 is some kind of exercise sensing protein in the endothelium. And as you know, of course, hominins have evolved over millions of years. This started Ardipithecus from 5 million years ago, and a lot of the time it's been a tough life. Yeah, no doubt, yeah. And our way of living now, as we're having to sit down like this, etc., is like being a fish or a shark out of water. This is not one of my sons, yeah, but this is more common now. So we all know that this is actually a challenge for us. And if you want to find a way to protect against cardiovascular disease and actually many other diseases like cancer, etc., then exercise, we know, is a fantastic way to do that. It's really a wonder kind of drug, but it's not a drug. Yeah, almost no side effects, and strongly protects you against cardiovascular disease. So we're interested in how this the system works and whether you can do something with it or not. And of course, We got quite an interesting reaction in the press about this. This is the Sun newspaper, so for those who are not from the UK, you'll know that this is not probably top ranking. Yeah, so we really hit the highlights here. Yeah, so they like a shocking kind of headline, and there was some other magazine, FM magazine or something, they said, never need to go to the gym again, ever. Well, of course, exercise is good for us, and we should all be exercising as much as possible, but what we're interested in, as cardiovascular biologists, is that often people have to have an operation, or they've got arthritis or something, and they can't exercise. Yeah, there are lots of people in society who can't exercise. And often the doctors are pushing people out of hospital, not to save money, but actually to get them to be physically fit again, because it's good for their health. So we're interested in whether you might be able to understand this system chemically, and therefore somehow enhance the exercise system, the benefits of it. But of course it's a complicated system, but maybe you can get some health benefits from this. So that's the idea, is whether you might be able to have a small molecule modulation. And this was actually a fantastic study, not done by us, but done by Novartis and Ardem Pataputian, published 2005. So they screened more than 3 million compounds against piezo-1, piezo-2 overexpressing cells, just to look for an activator. Really a wonderful thing to do. And they did find a compound, and they called it Yoda-1, rather nicely. May the force be with you. So yeah, we've been interested in this compound, and of course many small molecules do all kinds of dirty things and things, but actually we think this could be quite interesting. So these are some cells that we've produced. So the tetracycline-inducible cells overexpressing piezo-1, so you can see the piezo-1 protein here. This is the Yoda-1 small molecule. In the non-expressing cells, there's no calcium response, but when you express piezo-1, you get a nice response, so that works well. And these are the knockout experiments. So here, I won't go through all the experiments, but we've isolated endothelial cells from the piezo-1 endothelial deletion. We can see a nice Yoda response in these endothelial cells, and in the knockout, there's nothing at all. So here, in the native cell system, then it's a piezo-1 dependent response, and other things are unchanged in these mice. So just the system technically seems to work quite well. I showed you before that there's a flow-mediated depolarization, which is lost in the piezo knockout. Yoda-1 very nicely mimics that flow-mediated constrictor effect, and in the piezo knockout, nothing. So that looks quite promising. So superficially here, using a small molecule, you can mimic the flow effect. These studies are not published by us, but what I'm just doing here is suggesting that Yoda-1 might be acting directly on the channel. This is from Adam's group. This is piezo-1 in lipid bilayers activated by piezo-1. And then Beilong Xiao's group recently published some surface plasmon resonance suggesting that Yoda-1 acts directly on the channel, perhaps around about here in this area, somewhere there. Well, we've been interested in the chemistry of this and whether this is actually some sort of real pharmacology and whether you can do some structure-activity relationships. And the first thing, we just published the first part of this here. I'll show you that the right-hand part of the molecule with these two chlorines is very, very important for the action of the compound. So just show you a little bit of data here for number 2E here, where we've lost those two chlorines, and then we've lost the activity completely. So here, where we've got one chlorine and one fluorine here, and the 2G, you can still get some activity, but in many cases, for example, with a double fluorine here, or just one chlorine here, then there's no activity. So this seems to be really critical. We then messed around a little bit with the pyrazine ring over on the left-hand side, and you can tolerate some alterations here. So show you for the 7A compound, that's still active, even though we've lost one of the nitrogens, and some other compounds here. So there seems to be more flexibility on the left-hand side of the molecule. We've also replaced the sulfur here with an oxygen, and you can still get activity as shown by this 2J compound here. But then when we've made further change, not only to this, so we've lost the sulfur here and put an oxygen instead, and we made further change to the pyrazine ring here, then we lost activity altogether. But we've still got the dichloro here. So we're actually interested in this compound here. And so this is this compound called 2K. We've still got the two chlorines here, but the left-hand side of the molecule is somewhat different. And this compound does not activate the channels, but it competes with Yoda-1. So this suggests that this part of the structure is necessary for binding, but not for agonist activity in very simple terms. And then so what we find is actually a reasonable competitive kind of curve. We've looked also using a thallium assay at spontaneous activity of the channels, which is shown here, and that's unaffected by this compound 2K. But if we activate the channels first with Yoda, we can get a nice increase in thallium entry through the channels, and then that's reduced by this compound 2K, which we've called Dooku-1, because now we're all into Star Wars, because this Count Dooku is a competitor of Yoda's, you see, so that's kind of where it comes from. There's also a compound from Beilong, he calls it Jedi, so no doubt this is going to be a popular thing. So this compound, Dooku-1, quite a nice competitor of Yoda-1, suggesting there may be some kind of specific site on the agonist site on the piezo channels that we can manipulate. This compound seems to be quite selective, no effect on the ATP response, TRPV4, TRPC channels, or operated entry, that kind of thing. And that's quite good. So I'll finish up there and just summarize by saying, first of all, I think piezo-1 in the endothelium, or more broadly, is a very interesting channel, and there might be some therapeutic possibilities. There's a lot more to learn yet. The piezo-1 is a critical sensor of blood flow. The flow sensing is rapid via a membrane-delimited mechanism, so it works in the outside-out patch. This protein is critical for murine embryonic vascular maturation, and without this you can't get vascular maturation or life in the mouse. Part of the signaling is through calpain and digestion of focal adhesions. If you allow the animal to develop normally, then you can study in the adult mouse. The mice are alive, but the protein's important in the endothelium for blood pressure and exercise performance. And we found in the endothelium the properties of the channel are a bit different from overexpressed channels, quite constitutively active. Also non-inactivating properties, but they are also fluid flow activated. And in the vasculature, they have an anti-EDH, hyperpolarization mechanism. So it's a depolarizing mechanism, actually a sodium-dependent mechanism. And there is some emerging small molecule pharmacology. So we think it's important in human physiology and disease. And there might be some potential for drug discovery one day. And thank you very much.
Time for maybe one or two quick questions.
So a very general thing about general pharmacology that I teach to medics. How does this local response slot in with the sympathetic vasoconstriction, vasodilation, circulating, locally innervated?
Yeah, well, we don't know that yet, but that'll be interesting to work out, yes.
Do you see activation in inside-out patches of these channels?
By flow. We have looked and we've seen nothing so far, so I think not actually, no.
And so you also mentioned that it's the depolarization caused by piezo-1 through gap junction that causes muscle to contract. So calcium flow through piezo-1.
Also a good point. Yes. So there's actually a dichotomy presented by this kind of channel, because you've got calcium and you've got sodium. Yeah, so you've got a depolarizing effect. You've also got calcium, and calcium in the endothelium is well known to activate nitric oxide synthase. Yeah, so you also get nitric oxide production. So there's actually a balance of two effects occurring. How that plays out in vivo, we don't know yet, but of course I've shown you some in vivo data about blood pressure and exercise, and the vasoconstrictor effect is dominant in that situation. But it may be in other situations the nitric oxide calcium effect is dominant. I personally think in a situation where we're more like this, then the nitric oxide effect is probably more dominant. But when you're exercising, you switch over to the constrictor effect. But it's early days to say. Yeah.